Surrounded by water filtration systems, climate control, and modern antibiotics, it is easy to assume that we have beaten nature. Because we experience far fewer environmental hazards than our early ancestors did, many people believe that human evolution has stopped. This assumption relies on the idea that the human evolutionary tree is a completed project, that we are a static, finished product standing at the end of a long timeline. However, our DNA tells a different story. Deep within our cells, our biology is engaged in an active, ongoing arms race against a wide range of environmental pressures. Our genome operates as a pragmatic survival toolkit, deploying specific genetic strategies to maintain our health in the environments we inhabit right now. Realizing that evolution is ongoing shifts how we understand our own bodies, forcing us to view modern diets, newly emerging infectious diseases, and basic survival mechanisms as part of a continuing, endless process. The first strategy our species uses is borrowing. Rather than waiting for a slow, natural mutation to emerge, early humans acquired traits already adapted to local conditions by interbreeding with archaic human species. Molecular evidence proves this occurred. Researchers recently extracted biological information from the enamel of 400,000-year-old teeth excavated from archaeological sites in China. This timeline maps a tiny amino acid variation. The data reveals Homo erectus passed this genetic variant directly to Denisovans. As these populations moved southward, Denisovans passed this exact marker into the ancestors of modern Southeast Asians and Oceanians. This ancient genetic legacy remains detectable in living populations today, persisting in the genomes of people in the Philippines and Papua New Guinea. Our DNA functions like a braided river, with different channels of humanity continuously mixing. Early humans utilized interbreeding as a rapid deployment strategy to survive in harsh, unfamiliar environments. The second strategy relies on spontaneous genetic mutations that make new food sources accessible, a phenomenon that accelerated as humans transitioned to farming and animal domestication. Historically, humans physically could not digest milk after infancy. The body naturally stopped producing lactase, the enzyme required to process milk sugars, leading to severe illness in adults who consumed it. Between 5,000 and 10,000 years ago, a genetic mutation emerged that kept the lactase enzyme active throughout adulthood. This occurred during a period of extreme environmental stress in Europe. Amidst famine and low sun exposure, the calories and vitamin D in cow's milk provided a massive biological advantage to those who could digest it. A single molecular shift converted a biological toxin into a vital resource, encoded into the biology of over 70% of adults of European descent. The third strategy involves the deactivation of our own genetic machinery to survive invisible predators like infectious diseases. The caspase-12 gene, which manages immune responses, contains a dangerous flaw. A fully functioning version makes the human body highly susceptible to fatal sepsis during bacterial infections. This cellular model compares a fully functioning sequence on the left, which triggers a cascading sepsis failure, with a deactivated sequence on the right. Human populations have gradually deactivated the caspase-12 gene, a move that increases the survival rate for sepsis threefold. This same strategy is visible in the ongoing fight against the HIV virus. This cellular model shows a CCR5 mutation acting as a physical barrier on the cell surface, preventing the HIV virus from attaching. This exact mutation scales back thousands of years, persisting because it protected our ancestors against a different ancient viral threat. Within the human genome, a disabled gene functions as a ruthless and highly effective defense mechanism against pathogens. Humanity's current survival edge consists of microscopic broken genes, mutated enzymes, and ancient proteins circulating in our bloodstream. Modern medicine and civilization shifted our selection pressures rather than halting our evolution. When humanity faces a novel threat, whether a dietary shift or a new pathogen, our genome actively responds by rewriting our biology. We are the latest iteration of a species that continues to adapt, functioning as a continuously updated survival machine.